2020, when the pandemic was starting to spread rapidly across the world, J&J felt the call to action. Our entire company rallied behind our vaccine team as we raced to develop a vaccine. At that point, there were no vaccines for COVID-19 and no treatments available. But within 15 months, the world would have access to a single-dose J&J COVID-19 vaccine. Our presentation today is the story of how J&J successfully developed the world's first single-dose COVID-19 vaccine in record time, changing not only how we approach vaccine development from a scientific and data science perspective, but also how we innovate and collaborate to deliver what was previously thought to be impossible. Vaccine development is traditionally a long process. Historically, it has taken upwards of 10 years to develop a vaccine from start to finish. But when the pandemic emerged, we knew that the world could not wait that long. We needed to accelerate the development of effective and a safe vaccine to provide a way out of this global crisis. We were able to condense a decade-long process into just over a year by taking several innovative approaches. First, we used our adenovirus 26 platform to create the vaccine. This is a platform we had invested in for decades, and we had honed through the development of our Ebola and Zika vaccines. Second, using highly innovative AI, machine learning, and data science-driven approaches, we were able to bring our large-scale phase 3 trials to the right COVID-19 hotspots at the right time, thereby accelerating the completion. Phase 3 trials are the large-scale human testing needed to establish robust efficacy and safety data for a vaccine. And in this case, proving that the vaccine is able to prevent moderate to severe COVID-19 disease in tens and thousands of participants. But how does it actually work? We start by taking healthy participants and splitting them into two blinded groups. One group receives the active vaccine injection, and then the other group receives the placebo injection. Vaccines in phase 3 trials need time to show whether they're effective or not. This is for two reasons. First, vaccines take a little bit of time to become effective in our bodies, to induce that immune response that protects us from future exposure to a disease. And then the second reason is that we need participants to potentially become exposed to this virus after they've been vaccinated to know whether this vaccine works. Now recall, these two groups are blinded, which means we don't know which group actually received a placebo or an active vaccine injection. For us to know whether or not this vaccine is effective, we now have to wait until a certain threshold of COVID-19 cases are reached in the two groups. We then unblind the groups and then examine the distribution of the cases between the vaccine and placebo groups. Now, ideally, if a vaccine is effective, you would see more cases in the placebo group than in the vaccine group. And that's that ratio that helps us determine our vaccine's effectiveness and efficacy. The key point is that it takes time after a participant is vaccinated with either the active vaccine candidate or the placebo to be exposed to the disease and for us to determine the vaccine efficacy. The time that a participant needs to wait translates to a long overall trial and how long the trial duration will actually be. So how can we decrease this waiting period? There are three options to shorten the time frame of a trial. One is your sample size. Second is the effectiveness and efficacy of the vaccine. And third is by setting up your phase 3 trial in locations with as high incidence of the disease as possible. And this last point is where the MIT Janssen collaboration focused on. I will now turn it over to Dr. Bertemis, who will discuss how our teams innovated together to solve this challenging problem by taking advances in data science and applying them to life sciences.
In order to model the future spread of COVID-19, we developed the Delphi Model, which stands for differential equations lead to predictions for hospitalizations and infections. We started the effort in mid-March 2020 when MIT closed for the pandemic. There is significant complexity in modeling COVID-19. Reliable data is scarce, and even data from the same sources can be inconsistent. There has been unprecedented societal interventions that greatly change the future spread of the pandemic. And on top of this, there are constant epidemiological changes, including new variants, treatments, testing regimes, etc. If we look to the graph, it is perhaps not surprising that when the New York Times reported on 5 models of the future of US COVID-19 deaths back in April 2020, the 5 models shown here showed very different curves for the future. IHME predicted a fast return to normal, while our model and Columbia's model predicted a slow decline. And Imperial's model suggested further exponential growth. The reality turned out to be the gray curve shown here, and I dare say that we did well. I would like now to describe how we went about to overcome these challenges to model the COVID-19 pandemic. There are 3 main characteristics of Delphi that directly address the previous challenges. First, Delphi builds off of classical compartmental models so that parameters are interpretable and require less data to produce reasonable results. In particular, the parameters mean that we can discover data consistency issues very quickly. Next, on top of interpretability, we introduce a data-driven modeling of societal response to explicitly capture the effect of the significant interventions during the pandemic. And third, we extended the epidemiological model with carefully chosen time-varying parameters to recognize the changing epidemiology. To build Delphi, we first start from a classic interpretable compartmental model, the SEIR model. This model separates people into 4 states based on their epidemiological characteristics: susceptible to the disease, exposed to the disease, infected by the disease, or recovered from the disease. Then it models the transitions between the states with ordinary differential equations shown here. The one that is most critical to its success is the equation connecting S and I, so we will look into it more closely. The differential equation connecting S and I can be decomposed as follows. The rate of people getting infected per unit time, say we choose per day, is equal to the infection rate alpha, interpreted as the number of contacts by each infected person per day, multiplied by the infection population, which gives us the total number of contacts by the infection population. And since there is S over n% of the population that are susceptible to the disease, the number of new infections is thus alpha multiplied by I and multiplied by S over n. This very simple yet effective modeling captures the first-order dynamics of disease transmission well and has resulted in its great success in many previous epidemics. However, there is one key assumption in this model that is far from true in the COVID-19 pandemic, which is the alpha value being independent of time. Given the number of societal interventions and behavioral changes, the alpha value is constantly changing in the pandemic. To remedy this, we introduce a multiplicative effect, gamma of t, which measures how much the natural infection rate, alpha, is reduced by the total effect of societal interventions. So if gamma of t equals 0.3, that means at time t, the infection rate is reduced to 30% of its natural value. To specify gamma of t, we assumed that the interventions can have 4 phases. In phase 1, the interventions start to affect the pandemic spread, but slowly. As the pandemic progresses, in phase 2, the response would slowly increase in intensity, pushing gamma of t to decrease faster until it reaches some saturation point in phase 3. In phase 4, there might be a potential resurgence due to early relaxation of government interventions. We chose a parametric form for gamma of t, using an arctank modeling to capture the concave convex part of the curve and the exponential part of the resurgence. The parameters highlighted control the length, intensity, and magnitude of each phase and are optimized with respect to the historical cases and deaths data of each region that we applied Delphi to. So, different regions would be on different parts of this curve based on their recent epidemiological characteristics. One particular modeling choice we would like to explain more deeply is the selection of the arctan function. There is a large class of functions that can be used to model a concave convex behavior, and arguably the sigmoid function might be a more natural choice. However, one key observation is that the decay rate of the response gamma of t controls the length of the epidemic before any resurgence. The sigmoid function decays quickly, and thus the resulting epidemic is short. The R-time function induces an inverse relationship between gamma of t and time, which permits a much more prolonged epidemic. Experimentally, this modeled the long tail behavior in the 2014-2015 Ebola pandemic well, which was one of the most recent major epidemics that resulted in significant societal intervention. Of course, government interventions are only one of the many key effects in the pandemic that we modeled. Other key considerations include additional states to account for underdetection of cases, as well as utilizing adaptive mortality rates and independent recovery/death processes to account for the great difference between patients who eventually survive or die. Recent additions further include consideration of vaccinations and variants. With all these additions, here we show the full diagram of the Delphi model. We see the separation of the undetected and the detected cases, and we further divide the detected cases to those quarantined and hospitalized. We also observe in this diagram separate processes that model patients who eventually recover or die. Now, with all the additions, the final model output is highly non-convex in the input. In total, Delphi has 11 tunable parameters, including those that control the societal interventions, probability of death, etc. To optimize Delphi effectively with respect to the historical cases and deaths, we utilize optimization to balance rapid local updates and global optimization. Specifically, we utilize the truncated Newton method for daily updates and use the global dual annealing algorithm weekly to correct for a new global minimum. The detailed process is shown here. We are plotting the objective loss against parameters. Say we start from the black dot, then using the truncated Newton method, we would search over a small region of the neighborhood and optimize greedily. We would then repeat this process on a daily basis to provide efficient local updates that allow the Delphi model to respond to new daily information quickly. Then, when we reach the point to run our scheduled dual annealing algorithm, we optimize over a larger range to detect new minima caused by potential pandemic behavior change. To ensure that this local minimum is truly representative of future performance and not just due to overfitting, we compare the model trained by dual annealing with a standard truncated Newton method on a holdout validation set and take the best performing model. So, how did Delphi perform? We observe that Delphi consistently performs well across regions and time. The graphs here show the Delphi predictions during the first wave of the pandemic for the United Kingdom and Russia. As one can see, we were very accurate in predicting the almost straight line type of growth in cases in the United Kingdom almost 1 month in advance. What is more striking is that we predicted the exponential worsening in Russia on April 17th, when Russia only had around 20,000 confirmed cases. We correctly identified both the speed and the magnitude of the exponential worsening. At that time, our predictions were so controversial that we got a few angry emails from Russian website visitors who claimed we were scaremongering. Well, I wish we were just scaring them. The good performance of Delphi extends to further waves. During the second wave, we compared our model to other top models submitted to the CDC on the hardest metric that CDC records, 4 weeks ahead death predictions. As we can see in the table, Delphi is consistently the best performing model throughout with an average rank of 1.77. The graph further breaks down the rank by week, with the Delphi model in the solid blue line. As we see, the Delphi accuracy rank never drops below average and is the most common leader of the board. Given the mounting difficulties of long-term prediction, we came up with a new idea. Instead of trying to predict what the epidemiology is going to look like months in advance, can we consider a range of societal responses and then robustly optimize the minimum possible incidence? We consider a range of possible responses and predict the pandemic under each of these possible futures. Then we choose trial sites that achieve a target minimum incidence under all scenarios. That way, when reality eventually occurs, we would have confidence that it would reach the target minimum incidence. To do this, however, we would need to know how the pandemic would evolve under different societal responses. And this is where the data-driven modeling of societal responses in Delphi comes in handy. Remember, in the Delphi model, we have a parametric intervention effect term gamma of t. For a given region that we fitted Delphi to, we can map gamma of t to the actual policy that was instituted in such area. In this way, we extract the average effect of each policy by averaging over every day such a policy is implemented. So we can model how the pandemic would change under different responses by changing gamma of t appropriately. Specifically, consider a scenario where we consider the government relaxing the restrictions from stay at home to only restrictions on mass gatherings and schools. In the bottom left, we plot gamma of t, which measures the effect of intervention, up to the change time. Then, at change time, we assume that gamma of t increases instantly with a magnitude that is proportional to the difference in the historical average under each policy. The proportionality constant is chosen to ensure normalization. Of course, in reality, a change in government intervention is hardly instantaneous, but we found such modeling simplification to not have significantly impacted our results. Then, following such change, gamma of t continues its natural evolution into the future, now with this extra constant increase. This new gamma of t then would allow us to simulate how the infected cases would behave as a function of the scenario and time. With the number of infections in each region as a function of the scenario and time modeled, we can now formally present our trial location optimization procedure. The decision variable is whether or not we would select a region as our trial site. Our goal is to maximize the average trial incidence for the worst scenario, subject to the constraint that for each selected location, the incident for every scenario needs to always achieve a minimum value that is necessary for the trial to operate there. There are further constraints on locations, age, demographics, etc. In total, this optimization model robustly selects the best trial sites subject to a range of future interventions at each possible site. In the actual implementation, Janssen curated the uncertainty set for each region based on its political, social, and demographical characteristics.
In terms of implementation, I'd like us to think about it in 3 stages: country selection, site selection, and then site activation and recruitment acceleration. For country selection, we actually started our first readout in May 2020 when we selected our countries globally for a phase 3 trial for an anticipated September 2020 start. So why so long? Because it takes time to set up operational and regulatory considerations, such as health authority approval to conduct trials in their country, and supply chain logistic distribution issues. And so we had to make our country selection more than 3 months ahead of when we actually anticipated the trial to start recruiting. Before we began with our model usage, we were considering a wide range of countries. These were places where we had experience conducting other trials, but we didn't know whether COVID-19 would likely be high incidence in these locations or not without our model. For example, we were considering countries in Europe, such as the Netherlands, Germany, Belgium, or places in Asia, such as South Korea, Taiwan, or Japan, or Australia. And while some of these countries did actually experience an outbreak later in the year during our trial recruitment period, many of the countries, such as South Korea or Australia, had more limited or more controlled COVID-19 waves due to their government intervention during our late 2020 trial recruitment and case accrual period. That was why it was so important for us to model the impact of government intervention on the spread of COVID-19. In contrast, when using the model, the countries that we selected with its guidance included Brazil, Mexico, other parts of Latin America, and South Africa. These were all countries that then experienced significant waves of COVID-19 as predicted during our trial recruitment and case accrual period. Then in June and July 2020, we moved onto our site selection within each country, where we honed our predictions to the specific hospitals and trial sites that we wanted to conduct our trial in. This is where we really refined and evolved the Delphi model together with the MIT team. We added additional metrics such as age distribution, social compliance such as mask wearing, population density metrics, and in particular, we focused on diversity metrics such as racial and ethnic distribution at a county level within the US. This focus on diversity was critical for us to have a successful trial, and as a result, we ended up with the most diverse COVID-19 vaccine trial to date, with more than 19% Black and 45% Hispanic populations as an example. As we were refining the model, we also found that we needed to improve the data that we had. No one source had all the information that we needed. So we built a custom pipeline that pulled in the data from a variety of public health sources and created our own dashboard to specifically track COVID-19 incidents, cases, and deaths, updated on a daily basis. This daily updated dashboard was particularly useful as we then moved into the site activation and recruitment acceleration phase in September, October, and November 2020. During this stage, we needed to prioritize which sites to activate first and then which ones to really help support with maximally accelerated recruitment. The MIT Janssen model was critical for this stage because we needed to make rapid decisions on prioritization and be agile with where we thought the disease was going to be in the next few weeks. All of this culminated with our interim analysis in early 2020 that led to the successful U.S. Food and Drug Administration's emergency use authorization in February 2021 and European Medicines Agency in March 2021 for our single-dose COVID-19 vaccine. Now, to talk about the impact of all our efforts, I'll turn it back over to Najat.
Our machine learning model in partnership with MIT had significant impact on our phase 3 COVID vaccine trial. First, it was highly accurate in being able to predict the right hotspots 3 to 4 months in advance at a country, state, province, and county level. And this high precision had huge impact in 4 dimensions. Number 1, to actually shave off about 8 weeks of our development timeline. In a pandemic, every day counts. Second, we had a highly diverse trial, reflective of the population that we would vaccinate. Number 3, we actually had highly important clinical data on variants, which, as we can all imagine, is important as the pandemic continues. And number 4, it also helped us to learn how to develop these machine learning models to accelerate trials in COVID, but also beyond COVID, which we're now applying across our Janssen pipeline. But the real impact is not measured by just the success of the trial. It's measured by the significant individual, societal, and scientific impact that the acceleration of our vaccine to the world was able to achieve. I will talk about this impact across 4 dimensions. First, the directly vaccinated individuals and their health outcomes. How many hospitalization, ICU stays, and deaths were directly able to be avoided due to the acceleration of our vaccine? Second, close contacts of directly vaccinated individuals and their health outcomes. The adjacent effect of preventing the spread of the disease because of our vaccinations. And third, the economic benefits in the reduction of medical costs and preserving economic productivity. And last, the additional scientific insights that we were able to gain as a direct result of using the model to suggest sites in areas of extremely high incidence of the disease. Finally, we'll talk about the global impact of our vaccine. The fact that we have been able to reach out to more than 100 countries, many of them low and middle income. And we will speak to the implications of our success, how this experience has transformed the way we use AI, machine learning, and data science to drive our drug development. Accelerating emergency use authorization allowed our vaccines to reach millions of people earlier and directly enable extraordinary health outcomes. In the approximately 6 to 8 weeks that we saved during our phase 3 trial, we estimate that about 8 million people in the US were vaccinated. This translates to more than 7,000 hospitalizations, 1,800 ICUs avoided, and more than 1,100 lives saved. When we look at the acceleration and the impact across the entire globe, it led to 9.5 million vaccinations administered earlier, more than 9,000 hospitalizations that were avoided, and more than 2,000 ICU stays averted, and almost 1,400 lives saved. Every single life matters. But the power of vaccination is that it not only prevents severe outcomes in those directly vaccinated, but it also helps protect the family and friends of vaccinated individuals via a more adjacent effect. Based on studies to date, each vaccination has helped prevent an incremental 1.36 new cases. Now, what that means is that in addition to the millions of lives impacted directly, we also indirectly help prevent an additional 13 million new cases, 12,000 new hospitalization, 3,000 ICU stays, and more than 2,000 deaths in the family members and those close to those that were directly vaccinated. As you see in the animation, the health outcomes enabled by our acceleration of the vaccine also translate to economic benefit. There are direct savings in avoidance of adverse health outcomes, the cost of treating a person in the hospital or ICU, and ultimately the estimated economic value of their lives. There are also productivity savings, which is preserved through the avoidance of work loss due to moderate to severe COVID-19 disease. From those that were directly vaccinated, we estimate about $455 million in adverse health outcome savings and $340 million in productivity savings, for a combined of almost $800 million in economic benefit. And for the close contacts and family members, we estimate that saving to be over $1 billion. The total economic benefit from our accelerated vaccine is estimated to be close to $2 billion, about $1.88 billion globally. And then the last piece I want to touch on is that the MIT Janssen model also helped us to go to local sites in Brazil and South Africa due to the extremely high incidence of COVID-19, all unbiased decision-making, just focused on where the data was. These countries have also generated some of the most disruptive new variants. For example, the P1 variant, named Gamma by WHO, first emerged in late 2020 in Brazil, in a region that we predicted to become a hotspot. The Beta and Omicron variants are both variants that were identified in the southern regions of South Africa, including South Africa in the Gauteng region, where Omicron rapidly spread. The model had predicted these areas as hotspots, and the fact that we were there in these hotspots with clinical trial sites helped us to generate really valuable clinical data. Finally, and critically, the acceleration of the J&J vaccine has enabled earlier progress against our commitment of having global impact, including low to middle income countries. In many of these countries, it's difficult to store the mRNA vaccines due to the need of advanced refrigeration and the need to schedule and coordinate multiple doses to achieve protection against severe outcomes. The J&J vaccine remains the only single-dose vaccine granted EUA, and we've been able to make our product available over 100 countries around the world. The pandemic will not end if we don't address this issue globally, not only for epidemiology reasons, but also for the imperative to increase health equity and access to clinical medicines like our vaccine across the world.
Let us address the transferability of the work in other domains. My group at MIT developed the covidanalytics.io website, which is active today. During the last 2 years, it has had about 10,000 visitors per day from all over the world. The website includes the Delphi Model predictions for 120 countries and all US states, as well as a variety of other tools, such as optimizing the distribution of ventilators, triaging COVID patients for hospitals, among several others. A visitor of the website can experiment with different intervention policies such as lockdown, no restriction, restrict mass gatherings, among others. The Hartford healthcare system utilized the Delphi model to size its ICU and staff capacity.
We have a 2,474 beds acute care facility in Hartford Healthcare. We also have over 160 critical care beds. And one of the challenges we had was to expand the number of critical care beds in the pandemic. And our clinicians worked with Dimitri's team to understand how the Delphi model can assist in managing the surge or the stress we're about to face with the pandemic. Essentially, what the model provided us is a projection model, projection of our strain on our capacity, especially the beds, ICU, critical care, or our staffing as well. With the help of Delphi model and assistance from professor and all the resources MIT provides, we were able to navigate the pandemic with obviously some difficulty, but definitely with ease, given the prediction model was assisting us at that time.
Delphi was also utilized in an important effort to optimize COVID-19 vaccine allocation in the United States. This project dates back to early January 2021, when the new US administration announced a plan to vaccinate 100 million people in 100 days. As part of the plan, FEMA planned to open 100 mass vaccination centers around the country to direct the federal-led effort and accelerate the vaccine rollout. Given the limited vaccine supply and the large variance in epidemic dynamics across the country, it was key to allocate the centers efficiently to save lives and achieve the maximum effect. Therefore, our goal, working with FEMA, was to optimize the locations of mass vaccination sites in the US to accelerate the end of the pandemic. To do this, we created our optimization framework as followed. We first decided where to locate the mass vaccination centers. At this level, we prioritized hotspot locations while ensuring nationwide equity. Then, we decided how many vaccines to distribute in each center every day, where we dynamically updated our allocation as the pandemic changed over time. Following that, we decided who to vaccinate in each center, where we prioritized vulnerable populations while also ensuring that locally within states, the centers were allocated equitably. Then finally, after we decided all of these layers, we then leveraged the Delphi model to accurately simulate the infection dynamics in each state and predict the final death toll. We then optimized this multi-layer decision process to minimize the Delphi-driven death toll prediction. The results of the Delphi-driven vaccination plan can be seen on this graph. The shading shows how many lives could be saved using the Delphi optimized plan compared with FEMA's original allocation. In total, the optimized plan saves over 4,000 lives in a 3-month period. Given such outsized impact, FEMA opened mass vaccination sites according to our recommendations and utilized our high-level strategic insights to prioritize states most in need, including Florida, Missouri, and Texas. This implementation eventually led to thousands of lives saved. Given the adaptive nature of the Delphi model, we were able to utilize the Delphi-driven optimization to tactically allocate vaccines to each state on the day-to-day level. Our efforts were officially recognized by the White House, and we received a presidential seal as a commendation.
You've heard from Dimitri and Michael on the fantastic technical solution that they developed to help predict where the hotspots for COVID-19 were going to be. At Janssen, we realized that in order to utilize these predictions, we had to work closely with the MIT team to refine and evolve them even further. In fact, from the start of our collaboration in April of 2020, we were working together day and night, quite literally day and night, every day, to be able to come to the right answer for our phase 3 trial. And as you see on this mosaic in the screen, there were so many members who were contributing from all places across the world to be able to develop the solution together. I'll turn it over now to Michael to talk a little bit about how we work together and really refine the model for our purposes. Michael?
As Jenny's mentioned, we had daily meetings with stakeholders across the Janssen organization to analyze the latest results from the Delphi model in response to the rapidly changing pandemic situation. and also iteratively update to better model behavior in certain key target areas that Janssen was looking at. In particular, we also created a scenario analysis toolkit to help Janssen analyze the potential future pandemic situation across a wide variety of scenarios to allow robust planning for the upcoming trial. I'll hand it over to Dimitri.
So this has been one of the most intense as well as exciting collaborations I have had in my career. I have, from April 1st, 2020 until the end of the summer, we have literally met easily 5 times a week for 1 or 2 hours per day, talking about new trends, new ideas at a level of exchange that in addition to the hour or hour and a half we met, continued during the day. Of course, it has been very adaptive, and I'm quite excited about what we have achieved.
As you've heard through this presentation and from my colleagues and our partners at MIT and Harvard Healthcare, data science played a crucial role in the development of Johnson & Johnson's COVID vaccine. We knew that placing our clinical trial sites in hotspots was necessary if we wanted to see the efficacy of our vaccine as quickly as possible. If people weren't at risk of being exposed to COVID, how would we know if our vaccine was effective? The fact that our team was able to predict those hotspots with 90% accuracy had huge impact. We talked about how this enabled us to accelerate our vaccine development timeline by 6 to 8 weeks. Not only was this an impressive feat from an operational standpoint, but more importantly, it enabled our vaccine to start reaching patients in need 6 to 8 weeks faster at a time when 10,000 people a day were dying. Our work translated to infections averted, lives saved, and families whose loved ones are still at their dinner tables. These efforts also enabled us to execute one of the most diverse clinical trials, reflecting the populations most at risk. The importance of this and the example it sets for the entire industry cannot be underestimated. What I want to emphasize is what it took to get here. The grit and determination of the collective team and the willingness of leaders to take risks and do what was right at a time when the whole world was watching. The easy thing to do would have been to simply place our clinical trials at sites where we'd worked before, but our team doesn't do easy. When our data science team came forward with a proposal to leverage artificial intelligence and machine learning to aid in site selection, our leaders didn't say it would be too difficult or that it couldn't be done. They listened and they took a chance. This, to me, is a critical inflection point for our industry. Gone is the era of doing things how they've always been done. Because when you see proof firsthand that there's a better way, it generates momentum. R&D in the future will look a lot different than it does now, in large part because of how data science is changing the game. At Johnson & Johnson, we're committed at the highest levels of our company to leverage the power of data science to fundamentally change the trajectory of human health, because we believe data science is just that powerful. What we learned from COVID-19 is acting as a catalyst across the entire portfolio. In addition to aiding with site selection and clinical trial efficiency, data science is enabling all sorts of things. We're using data science to better understand the diseases that we're working to combat. We're using it to select the most promising compounds to move into clinical development. We're using it to identify patients who could benefit from a more targeted treatment and facilitate their recruitment into clinical trials. We're using it to aid in earlier and accurate diagnosis of patients suffering from rare or difficult to detect diseases, leading ultimately to better patient outcomes. The application of data science for our COVID-19 vaccine trial has opened up so much possibility for both how we innovate and impact patient lives. I truly believe that this is just the beginning of a data science revolution in healthcare, and I'm so excited to see what's to come and the impact it will bring. So on behalf of the entire Janssen team, it is an honor to be recognized among such outstanding work, and we thank you very much for considering us for this prestigious award.